Nick has been an entomologist and geneticist, has been researching honeybees for over half his life. Before joining WSU team, his doctoral work focused on genetics and behavior, where he documented how networks of genes in the honeybee brain turn on and off foraging and mating behavior. His current research seeks to find innovative ways to improve honeybee health. This includes use of fungi to create better bee food additives and using pathogenic fungi to kill varroa mites. In the laboratory, he uses genetic techniques to measure bee pathogen levels and test the effects of treatments on honeybee immune system. So, Nick, welcome to the show. You can share your screen whenever you like.
Okay, hopefully everybody sees the slides. I'm definitely a geneticist by training, but early on during my undergraduate years, I started working with the honeybee lab. That was at Ohio State at the time when Sue Kobe was there. So I was very fortunate to get to learn my basic beekeeping from kind of a world expert in bee breeding. And I really got hooked on bees, and I never lost that as I continued through my PhD doing a lot of work on genetics of bees. I used to do a lot more with what controls bee behavior and a lot more neuroscience and pure research, using bees the way other scientists use fruit flies or rats or so on. But after colony collapse disorder, I thought I really needed to shift gears and use my skills to help do some good to fix the bad bee situation. And that's what brought me to WSU. So today I'm going to talk about my lab's research using fungi to help save bees. And there's two main projects that we really do. So as a quick outline, the first one is about using fungal extracts to improve honeybee health. Basically, we have a system where we can grow fungi in the lab, make these extracts. This can be added to honeybee sugar syrup feed, and this makes it a lot healthier for them. It extends bee longevity, helps them fight diseases, and so on. And then our other big avenue of study is there's this Metarhizium fungus that is a pathogen to many different types of insects and arthropods. And we are developing strains that will directly kill the varroa mite. So first, I'll talk about bee nutrition and what bees are lacking, the problems facing bees with bee nutrition, and then what we're doing with fungi to help. And then at the end, we'll talk about killing mites with Metarhizium. So a little bit about just honeybee nutrition in general. They need a lot of the same nutrients that you and I do. So carbohydrates for energy, basic sugars, starches, and fats. They need amino acids and proteins to help build cells and do things in the body. They need some of the essential fatty acids. So again, like you and me, they need some of the omega-3 fatty acids, omega-6 fatty acids. There's a few that the bee can't synthesize themselves and they have to get it from their food. Vitamins A, B, and C, and then minerals, potassium, calcium, zinc, and so on. So with all of these are things that bees need. Their nutrition is a lot like ours in many ways, except that they have to get it, well, most of it from flowers as a resource. So pollen and nectar need to contain all these things that bees need. Well, unless the bees are foraging on fungi or other things like that. There's a few other nutrients that are not talked about much in the honeybee world, but I want to bring them up here. This one is sterols. These are the building blocks of hormones. So testosterone, estrogen, cortisol. You have several steroid hormones in your body. Honeybees will use it to make the hormones that they use to control different life stages. So the hormones that control the larval to pupal transition are made out of sterols. And again, bees cannot produce these on their own. They have to get them from their diet somewhere. This can be a plant or fungal source. It's sometimes in pollen, but we think that they might be foraging on fungi to get this nutrient as well. If you're more interested in more details on this, Ramesh Sagili's lab at Oregon State University has done a string of experiments with this, showing that sterol supplementation can help bees out. And I do hear of beekeepers putting sterols into their pollen patties, just another way to make sure that the bees get all the nutrients that they need. Another interesting thing that we've learned in just the last 10 years or so is that honeybees need, or are healthier anyway, when they have polyphenolic compounds in their diet. So these phenolic compounds, they're the thing that make blueberries blue and your raspberries red. They give a lot of fruits their colors, and they tend to be very healthy for people, and they also tend to be very healthy for bees. There's polyphenols that are found in nectars, honeys, and pollens. And what May Berenbaum's lab at the University of Illinois did was they went to test the effect on the bees' immune system and their health. So what they showed was first that when you feed bees honey, that their immune system is boosted in several ways. And the effect, one of the primary drivers of why honey is better for the bees than sugar water, is the presence of these polyphenolic compounds. So when you do give bees these polyphenolic compounds in their honey, or if it's given in their sugar water, the genes associated with detoxification of pesticides go up, and some of the genes involved with fighting pathogens go up as well. So it really seems that much like you eating your fruits and veggies, when bees get these chemicals in their food, they end up better off. They end up healthier. They can fight diseases better. They can fight off pesticide pressure more easily. A little more about these phenolic compounds. They're a very diverse group of compounds. Many have antioxidant properties. If you think about the red wine ingredient resveratrol, or that's one of these polyphenolic compounds, or the chemical in green tea that has such good antioxidant properties, that's another polyphenol. Well, anyway, they contribute to the color and flavor of honey, and different ones that you get from different plant sources contribute to all the different colors and tastes that we get with honey. So bees are consuming a lot of the same chemicals that we get in our fruit or vegetable-based diet. And just like these antioxidants are good for you, they're good for the bees. They seem to make the bees healthier. Bees really like a diversity of diet. And I've already mentioned that these phenolic compounds, you can find in blueberries, some are in chocolate and so on. But the idea of sitting down and making a meal entirely out of blueberries or entirely out of chocolate isn't super appealing. There seems to be limits to how much of one type we want. And in fact, having a diversity of types of these compounds not only tastes better, but tends to be healthier for you. And we think this is exactly what's going on with bees. They want a diversity of nutritional sources to make sure that they get all of the vitamins that they need, all the essential amino acids. No one pollen or nectar is going to be perfect to provide everything that they want. They really want a diversity of foods just like we do. Now, one thing that we frequently overlook as beekeepers is that bees do forage on more than just flowers. So one of the ones that a lot of beekeepers have noticed is that bees love a stagnant, algae, scummy pond. They will fly past clean water sources to forage on scum-covered ponds. And in fact, there have been reports, in fact, even in the scientific literature, proven cases where the bees were foraging on algae. A couple of years ago, there was a fun paper from the Czech Republic where they thought the bees were foraging on green pollen. They couldn't figure out what it was. They finally did a genetic test and found it was algae. The bees were packing it on their legs and bringing it back to the hive. Algae is not super nutritious for them, but it does provide some vitamins and minerals and so on for the bees. Tree saps. This is one we're typically more familiar with because we've all had to deal with propolis. So bees will forage on tree sap, bring it back to the hive, and turn it into propolis. Propolis contains many of these phenolic compounds, has a lot of antifungal, antimicrobial properties, and is generally good for the bees to forage on it and bring it back to the hive. Helps protect the hive from chalk brood and some other things. Honeydew, also frequently forgotten. Here's a bee foraging on honeydew that's been excreted by aphids. When aphids feed, they're fairly sloppy feeders, and a tree sap-like substance that they couldn't fully filter ends up all over where they are feeding. Bees will definitely forage on that and turn it into honey. In fact, if you go to places like Turkey, they really prize their pine honey, which seems absurd because pines don't make flowers. But what it is the pine trees have sap-sucking insects, and there's so many of them that bees can actually collect enough honeydew to make honey from it. And so you can, in fact, get pine honey is just coming out of the back end of an aphid instead of out of a flower. Cow manure. This shocked me when I first saw this in Illinois. What it probably has to do with is minerals. So in both the cow pie and mud puddle situation, typically what you see is the mud or the manure dries out, and then after a rain, there would be little puddles, in the mud or on the manure, and that will leach salts, minerals into that water. And the bees forage on that water, and they can get, especially some of the earthier minerals like zinc and magnesium out of it that way. So bees do forage on many different things, in addition to flowers, but for interest of this talk, we'll talk about bees naturally foraging on fungi. And as I've been doing this research and giving these talks, it's really quite common for people to tell me their experiences with finding their bees foraging on their oyster logs, their mulch beds, places where they are farming fungus or where fungus might be growing anyway. This is one from Eastern Washington where somebody was growing turkey tails and captured a bee in perfect mid-flight there. This is from Paul Stamets on the west side of the state also in a King Stropharia mushroom bed. We were talking about types of mushrooms bees like to forage on before the talk started. This one's from a King Stropharia. Bees do like it. This is from Maryland, USDA. Jay Edens, probably one of the top bee researchers at the USDA, sent me this picture of bees foraging on the rotting, crumbling log in their apiary. This one's from India. It's really fun. This is growing out of somebody's house, and this is actually not a honeybee. This appears to be a leafcutter bee of some kind. Here's another one I got off the internet. And then there's a great paper a couple of years ago from Japan documenting the Japanese honeybee foraging on these bolete mushrooms. So it looks like the Japanese bees like their porcinis. Well, it's a little surprising perhaps that bees forage on fungi, but when we look a little closer, perhaps it's not so surprising. Fungi are actually part of the bee's diet in several different ways. The one that's perhaps the most common is when bees bring in pollen, they process it into bee bread. The primary function of this is preservation. Pollen can begin to mold and go bad after a few days, but bee bread can last them from September until March when they raise the first of the spring brood. So they do this mainly for preservation purposes, to make the pollen last longer. And there are beneficial fungi that do aid in this fermentation process that the bees do to turn the pollen into bee bread. It's most analogous perhaps to pickling because it's an acidic fermentation that the bees do. The fungi involved include Aspergillus, Penicillium, where we get penicillin, the antibiotic, and Cladosporium. These are fairly common fungi that are around. There's also bacteria involved, particularly the lactobacilli. So it's a bit like yogurt making also. But I do find it interesting that bees use many of the same microbes that we do for food preservation. They do for their pollen preservation as well. And yeah, it's not too different from the bees making sauerkraut to preserve the cabbage. They're pickling their pollen to make it last longer. Well, when they do this, there is a fundamental change in the nutrient profile. Overall, there's a loss of calories. The yeast and microbes eat up a lot of the sugars and so on, but there's a gain in other nutrients, particularly vitamins, sterols, and a few others. So bees do end up eating a lot of fungi as they eat the bee bread in their diet. Beekeepers also give their bees fungi quite a bit. It's easy to forget that yeast is a unicellular fungus. We typically don't think of yeast as a fungus because it never produces a mushroom. It doesn't even grow mycelium for that matter. It's unicellular, but it is a fungus, and the nutritional profile of yeast is not that different from what we get with other fungi. So when bees eat yeast, which is one of the most common things put into pollen patties, brewer's yeast or nutritional yeast, the bees get B vitamins, sterols, proteins, and some other nutrients. And one of the good things about this is we know that simply giving the bees a fungal-based supplement, that the bees will readily consume it. They process it quite well in their gut. They can handle eating fungi. They seem to be very well adapted to do so, whether they forage on it naturally or if the beekeeper provides it. Honeybee nutrition, it's not great for a lot of bees. It's so strange for me to hear you talk about the size of the brood ball that you have when we are under 5 inches of snow right now. Most bees are not fortunate enough to have access to good nutrition throughout most of the year. A lot of flowers bloom in the spring and summer, and then not a lot else after. Here are some pictures from the Pullman area. We get March and April wildflowers. We get some June wildflower bloom also, and then we hit the dry season. And it really is from July to the following March, there's nothing. There's very, very little for the bees to eat. And so they're under a lot of nutritional stress. They have to rely on their honey stores or what the beekeeper gives them just to survive. This was particularly bad this year with the drought and fire season that we had. And then beekeepers can sometimes make this worse by harvesting the honey that the bees do make in the spring and summer when everything's in bloom. And then there's nothing for the bees to eat in the fall, and they don't even have their honey to fall back on. When I used to live in Ohio, it was amazing the fall nectar flow we would get. There's a lot of old field mixed woodland areas, and we get these goldenrod and aster blooms. And it was remarkable. You could take every frame of honey out of a hive in late August, and they would rebuild 80 pounds of honey by the end of October. When I lived in Illinois, less so. Too many cornfields. There wasn't enough bloom. And now here in Pullman, there's no fall flow. So the bees really are nutritionally stressed most of the year. The migratory operations, the commercial pollination operations, those bees might have it even worse. They frequently end up in these holding yards, and clearly there's no food here at all. Then when an orchard does come into bloom, there's all the food that they could possibly want, but of only one type. And as we discussed, eating only blueberries or only chocolate for a meal, I really like almonds, but I cannot imagine eating almonds for a month with nothing else. And bees, wanting their diversity of diet, they don't like to eat only almond pollen for a month either. So in fact, it's very common. Several people have documented that when you plot bees in the middle of a full bloom almond orchard, a lot of those bees fly past all of those blooms to go for the wildflowers in the ditch farther away. They want that nutritional diversity, and they will fly to go get it. Not all flowers are the same. Well, the answer for a lot of these times of nutritional stress for beekeepers is sucrose feeding. White table sugar or high fructose corn syrup is also quite common, more in the commercial industry. Now, this has many, many benefits. It's how we keep our bees alive in Eastern Washington. And you can use sugar syrup very effectively to help boost your colonies in the spring and kick them into gear, as well as just, preventing them from dying. It has many positive effects, but it is not nectar. It's not the same as honey. And there's just many nutritional deficits that come from feeding sugar. Over here's some data from an interesting paper from Marcia Wheeler. She's now doing human genetic things, medicinal things for human benefit. But she used to work with honeybees, and she ran a very interesting genomic metabolic profile on bees fed honey, sucrose syrup, or high fructose corn syrup. This is a table of differences of gene expression in the fat body of the bee, basically a fingerprint of the metabolism of the bee. And what we find is that high fructose corn syrup versus sucrose, not that different. The bees' metabolism, their basic metabolic functioning, very similar between those two. This was done at a time when there was a bit of a hysteria about high fructose corn syrup being really bad for bees. It's not bad for bees, it's just not good. It's more of what it lacks than what it has. Well, anyway, if you compare any of these simple sugar syrups to honey, honey versus sucrose or honey versus high fructose corn syrup, there are major differences. So honey is fundamentally different in that it has these other things in it that the sugar syrups do not. Sugar syrups are deficient in many ways. Well, what are those many ways? Just about everything except for calories. So both sucrose syrup, your white table sugar, and high fructose corn syrup are almost completely devoid of other nutrients apart from the sugars. There's no proteins, no fats, no vitamins, no minerals. And this is remarkable because I would challenge you to find a natural foodstuff on the planet that doesn't have minerals. That is remarkable that there's such little nutrients in the sugar syrup. And of course, no sterols, no polyphenols or these other nutrients too. It's just calories for the bees. And yeah, it's deficient in many ways. And if you taste sugar syrup versus honey, honey tastes better. It's really clear that it's more nutritious. Even in taste, your body tells you that honey's better for you than sugar syrup. We all know this, but well, what do? Not every bee gets to eat their own honey. A lot of them are in these situations that I talked about where they have to be fed sugar syrup just to keep them alive, to keep them going, or to keep the migratory pollination operation going. This poor nutrition situation interacts with the other stressors that bees have, these other problems that are really affecting them right now. Nutritional stress makes bees more susceptible to pathogens. I would say there are enough papers on this. This is abundantly clear now that these bees under nutritional stress have deficits in their immune system and their ability to fight off viruses, nosema, varroa, many of these problems. There's also mounting evidence that it also makes bees more susceptible to pesticide damage. There's these interesting cases where they studied the effect of neonicotinoids in Europe, and they would put bees near canola fields treated with neonicotinoids. And the effect on the bees was very different depending on the region where it happened. And they think a chunk of that had to do with the other nutrition that was available to the bees at the time. If the bees had better nutrition, they were better able to deal with the pesticide load that came in with all that canola nectar. Well, all these factors interact in a way that is quite difficult for beekeepers, right? All these things, pesticides, the mites, the pathogens, and the poor nutrition, these are all going to shorten your bee's lifespan. And this is how you end up getting these population crashes where, your bees are doing great and then you come back a month later and something's kind of wrong and you come back a month later and they're mostly dead. It's, these interactions are shortening the bee lifespan. And if the hive puts all of these resources into raising a bee, and then that bee dies young and doesn't pay back the colony, well, your population just dwindles away. You never get the pile of dead bees. It's not clear what killed the hive because there's so many things working together. It's just your bees weren't healthy. They didn't live very long and the hive slowly died away. Well, or not so slowly died away. What can we do? Well, we can plant bee-friendly flowers, right? That's always one of the best things we can do, especially those that bloom in the late summer and fall. Around here in Pullman, I planted a lot of zinnias this year and watered them a lot to make sure that they kept blooming up to the first frost. However, a lot of bees are dependent on the sugar syrup. Maybe we can improve it. And so here we finally get to the main crux of my research, which is how can we improve the sugar syrup feed, particularly using fungi? Well, this original idea of feeding bees fungi and seeing how it affects their health comes from mycologist Paul Stamets. He was growing these outdoor mushroom beds and he noticed bees foraging on them with a a lot of vigor and fervor. He describes the bees turning over the mulch with he says it was like watching a human like turn over a truck to get what was underneath it. They were so eager to move the mulch aside to get to the mushroom mycelium that was underneath. In particular, what do the bees eat? Well, they might take some of the mycelium with them, but mostly what we think they do is that they're particularly interested in these exudates, these little liquid droplets that the mycelium produce. Here's microscopic pictures of mushroom mycelium, and we'll find these little droplets, sometimes called guttation. And these droplets contain all sorts of chemicals. It's a little controversial on what these things do. Some people argue that it's mushroom pee, it's urine, it's an excretion thing. The mushroom's trying to get rid of extra chemicals and stuff. Other people think it's a little more adaptive, that the mushrooms are putting antimicrobial compounds and so on to protect the environment around them from, pathogenic bacteria or other things like that. If you do ever just basically try these exudates, go and lick a mushroom that has them, they are not sweet. The bees are not foraging on this as if it were nectar. It's something else. They're getting something else from it when they do this. At the same time, when Paul was looking at this, he knew that some mushroom species, including some of the ones that he was growing, produce compounds with antimicrobial and antiviral activity. And it wasn't long after that he ended up reading that bees are having trouble with viruses. So at that point, he started contacting bee labs to see who would be interested in helping figure out what was going on. And so he did end up calling Steve Shepard at WSU, and they started up a collaboration to look at these sorts of things. The basic process for getting fungal extracts for honeybees, we have this great collaboration with Paul's company, Fungi Perfecti, and then the WSU bee program. Fungi Perfecti grows the fungus and produces the extracts. Here's one of their grow facilities. It's a fairly simple mushroom growing operation, fairly typical, except for maybe in the species that are being done. But little mushroom grow bags, and then the mushrooms fruit out of the top. Just, you see shelves and shelves of mushrooms being grown. They create these extracts, send it to me in the jugs, and I take those to the field, and I can feed bees sugar syrup with the extracts in the field, or more frequently, what I've been doing recently is going into the lab. Because we think there's these nutritional interactions that are going on, I've been doing a lot more with bees in these little solo cup cages in the lab, because I then get to control all the nutrition that those bees are getting, and I get a clearer picture of what the effect of the extracts are. So it's been a very fruitful collaboration that's been going on for about 4 or 5 years now. Well, overall, one of the most interesting things that we see is that when we give bees sugar water with extract, with mushroom extract in it, this extends their lifespan in cages in the lab. And this is fairly consistent. We've now seen it, repeatedly, 3 for 3. I would say 4 for 4, except we had a trial that we had an incubator failure and we had to throw it out. Well, anyway, here's some survivorship data, and just kind of going through the different groups that we were tested. Down here at the bottom, this blue line that had the most death, that was just sugar syrup. The next one up was birch. We grew these on birch wood, and we wanted to know if there's any effect of what the mushrooms were growing on as opposed to the mushroom itself. So we tested this birch control also, and it did not do well. Then we have the chaga mushroom, Anonotus obliquus. This is the agaricon mushroom, Fomitopsis officinalis or Larissiphomus officinalis. But the agaricon mushroom extract here, and then the amadou mushroom and Ganoderma lucidum, one of the reishis, did really quite well at extending bee longevity. Here is another trial we did later in the year with what were more like winter bees, ones that we expect to live a bit longer, and indeed their mortality didn't go down, as quickly. But we get a very similar order. Birch and sugar control did not do well. Chaga and agaricon are in the middle, and then amadou and our reishi, the Ganoderma lucidum, is again the winner. And so in both these trials, we had significant extension of bee lifespan by giving them extracts in their sugar water. One of the very interesting things that came out of this, well, there's this idea that many of the mushrooms produce these antiviral compounds. And indeed, these mushrooms have been tested and shown to be effective against many different types of viruses, including like HIV, HPV, many of the herpes viruses. There's entire clades of viruses that these seem fairly effective against. And this made us decide to go ahead and we look at viruses in bees and see what was going on, see if that was part of the lifespan extension. Well, so we did several trials here. I kind of want to apologize for what's a fairly noisy scientific graphic here. But what we have is we have 3 different trials, trial 2, trial 3 over here. For each one, we have a control cage, a set of control cages, I should say, that were fed only sugar water. And then these other ones were given birch extract, again, just the birch wood, amadou. This is an artist conch. Here's birch again with amadou, and then birch with Ganoderma and the turkey tail mushroom. Well, what we find with a couple different concentrations is that most of these, and even the birchwood on its own, we get a dose-dependent reduction in virus levels in these bees. This is for deformed wing virus, which, as many of you know, is quite problematic for bees. So anyway, these seem to help the bees reduce their virus levels. Birch sawdust has some effect on its own, but most of the extracts did better than that. We followed this up with an outdoor trial using 5-frame nukes. It was basically 4 frames of bees with a frame feeder, right? The long black bucket you can click in and fill it with sugar feed. Well, now that we have whole colonies rather than cages, we can do multiple tests. And so here we can test a colony at the beginning of the experiment and at the end of the experiment 12 days later. Here's a colony that just got sugar, and the virus level started here and ended here. Here is one that got the amadou mushroom extract. Virus level started here and ended down here. The reishi mushroom, Ganoderma resinaceum, started up here and ended down here. This is all for deformed wing virus. So the 2 extracts we tested had significant reductions in virus levels. Lake Sinai virus is an interesting one. When there's not a lot of brood in the colony, Lake Sinai virus tends to kind of dwindle down over time. And so even our control colonies in the fall is when we did this. These control colonies, they went down a little bit. It was actually an 82x reduction in virus levels naturally. But when we gave them the amadou mushroom, it went down to 7,000x. And then with the reishis, this is actually a log 2 scale here. It was a 3 million-fold reduction in virus levels. So it really does look like these things help bees fight viruses. And that really got us going in terms of, we need to figure out what's going on with these in terms of what exactly is in them that is helping make bees healthier and helping them fight viruses. If any of you are more interested in this research, this is a scientific publication. Paul Stamets and I are the first 2 authors. This is in Scientific Reports. We paid some extra money, so it is free sourced, open access. So it's easy enough to find if you just Google it. We've worked with Fungi Perfecti to try to finalize production methods. Fungi can be fairly finicky with what they are grown on, and the effect that you get at the end can depend on what the fungi was grown on. Birchwood was what we found to be really good, including this really big 500 colony experiment we did in California almonds. But the problem was that supply chains kind of ruled it out. I don't want to get too much into it, but what happened was in late 2017 and going through 2018, we had a whole string of inconsistent results. We were beginning to think, after we couldn't find other problems, that the original wood that the fungi was grown on, we were thinking that it had become infected with a different pathogenic fungus that was not good for bees. You know, many of these trees have all sorts of latent fungi growing in them. We thought maybe it was a toxicity thing. So we did a metagenomic survey of the wood that we were growing these on, only to find that basically the supplier had swindled us. they sold us much cheaper lindenwood sawdust instead of birchwood sawdust, which is about the third the price. We paid for birchwood and didn't get it, and the extracts that we produced over the course of the year had little to no effect. So that was extremely annoying. And now that birchwood, we really have trouble finding a good, consistent supply chain. Unfortunately, we've had to move on from that. So we at WSU have worked with Fungi Perfecti to find a replacement grow method. The one we're settling on that seems quite good is using alder, which is really common on the west side of Washington. This one we can get fairly easily, and it's not too distantly related to birch. The fungus is pretty happy. A little bit of oat and rice supplementation seems to help. We might have to do a little bit more to tweak it, but we think that we have the fungus growing happy and producing, extract with good outcomes for the bees, in a system that we can now scale up. It was really unfortunate that we did so much work with birchwood grown extracts only to find that we could only produce it in limited quantities. So this was a whole other year of work in the background, working with Fungi Perfecti to really kind of flesh out and finalize a good growth method that we can scale up. Yeah, we're doing some extra cage studies for the FDA right now, or some of them just finished up. With this most recent cage study that we did, we had already contacted the FDA because we were wanting to make this a FDA-approved bee feed. And the FDA said, okay, you've been doing a lot of comparisons to sugar water, and that's fine, but we want you to see the effect compared to honey also. We were always hesitant to include honey because we kind of thought there's no way any sort of anything added to sugar water is ever going to compete with honey. It's the bee's natural food. It's so good for them, that sort of way of thinking. But like, okay, FDA, we'll include a honey trial. It will be interesting to see just honey versus sugar water, what ends up happening. Well, in this most recent cage trial, sugar water and sugar water plus extracts outperformed honey. And this was really surprising. Our honey cages actually died off sooner than what we saw even in just sugar water. And then sugar water with plus extracts did the best. We don't really know exactly why. There could have been pesticides in the honey. There could have been other weird plant chemicals in the honey. Many beekeepers complain, that there are plants like privet, and some of these that produce some kind of weird dark honeys that is actually not that good for the bees. They forage on it, they store it, but it's not that great for them. And beekeepers in the Southeast US will very intentionally harvest all the privet honey away so the bees have something better to eat later on in the year. So we don't really know what exactly was in it, but one bit of the puzzle may have been we end up taking some of the bees that were dying in these cages and we looked under the microscope and found really high Nosema spore counts. I say really high, higher than what we found in the sugar water or the ones that had extracts. So maybe the honey inoculated the bees with Nosema spores. They very well could have been in there. We really don't know. Or maybe there's some other sort of interaction that made the bees more susceptible to Nosema, like pesticides in the honey, weird chemicals in the honey. We're not really sure, but surprisingly, sometimes these extracts or even sugar water can outperform honey in a way that I still find surprising, and I would like to know a lot more. We are planning some pesticide testing on the honey we used in these experiments, but this could end up being another argument in the end of why feeding bees sugar water with extracts might be good for them during these times of dearth, because even their natural food might not be that great. Some beekeepers are hesitant to feed honey back to bees because of disease issues and so on. In terms of the FDA registration, there are almost no FDA registered bee products. A lot of the ones that are out there are very much under the radar, including ones that are basically honeybee feed additives and should be handled by the FDA. FDA really hasn't bothered to get involved with the bee business. Well, but we're going to make them now because we want to do things the right way. Well, bees are generally considered livestock, and so they're grouped with chickens, cattle, and so on. The main thing that we're really facing in dealing with the FDA right now is they have a very important question, and that is, are these mushroom extracts a food or a drug for bees? Now, are fungal extracts food or medicine for bees? Many mushrooms produce antimicrobial compounds. This is from a Ganoderma mushroom, a paper on the different chemistry that they can do. I find this great because the first chemical they highlight that this mushroom produces, oxalic acid, a known miticide. So anyway, many different types of chemicals they can create, long fatty acid ones, phenolic compounds. They can do all this fun sugar chemistry, lots of different fun chemicals. And basically the reason for this is plants and fungi have a fundamentally different immune system from you or I. We mainly make antibodies to clear up infections. Plants and fungi don't do this. They do not have an adaptive immune system in that way. Rather than that, their immune system is a bunch of different antimicrobial chemicals. And right, as we discussed, bees can forage on tree sap, bring those antimicrobial chemicals back to the hive, and that can help protect it from chalk brood or so on. The mushroom group that showed the best effects in the bees so far is the Ganoderma genus, or the Reishi group. These have medicinal uses. So these have been used in traditional Chinese medicine for at least 2,000 years. There's a long history of medicinal use of this mushroom. And many labs around the world have investigated fungal compounds for antimicrobial, antiviral, and anti-cancer properties. There's definitely interesting chemistry going on, and undoubtedly there are certain chemicals in these extracts that we're giving bees that do have drug-like effects when they are concentrated. That being said, I could probably pull some chemicals out of broccoli that would have drug-like effects when concentrated. So this is an interesting thing where there are drug-like things in there, but is that really what's going on with the bees? Or are the fungal extracts food for bees? And this is the side that we're leaning on for a couple different reasons. We started to do a lot of nutritional testing on these fungal extracts to figure out what exactly was in them. And we do find that they have B vitamins, no pun there, B vitamins, some proteins and amino acids. They have sterols in them. Fungi produce a lot of sterols to solidify their cell membranes, and that is a perfect precursor for the bees to make their hormones and so on. There's also chitin components. One of the primary components of the insect exoskeleton is the same thing that fungi use to make their cell walls. And so they get these fundamental building blocks for their exoskeleton too. And then of course they have polyphenols that I've talked about previously. There's a fascinating thing that when there's an acid fermentation of wood, particularly of the wood chemical lignin, the fungi end up producing these polyphenolic compounds that are very similar and sometimes the same as what we find in nectars and pollens. These are the same ones that May Berenbaum's lab have shown to help boost the bee immune system. So we think these polyphenols are one of the things that's kind of going into the mix, to help make the bees healthier and help them fight diseases. Here, picture of honey with all the different colors that they can get. Here is my arsenal of fungal extracts with many different honey colors. Another interesting thing about the extracts is that the mineral composition in them is very similar to what bees get in their natural food. Bees' diet is a bit different from ours, and that is intensely enriched for potassium, phosphorus, and magnesium, and almost no sodium. Kind of remarkable in that way. So anyway, the fungus, the natural minerals that end up in the extract end up being acidic, pH of 3 to 4, much like much of the bee's food. They have these phenolic compounds, they have sterols, and they have this mineral profile also. It really is remarkable that almost accidentally, these fungi are producing a really excellent bee feed in multiple different ways. One kind of extra little bit, we have a couple collaborations where we've, to help figure out what exactly is going on with these fungal extracts. This is Dr. Michelle Flanagan at University of Montana. She was actually the one that first identified Lake Sinai virus, and she continues to do a lot of fun laboratory-based bee virus work. She had a cell culture system going. And this is very interesting because if you test viruses in bee cells in cell culture, this is outside of a nutritional effect and outside of an effect of the bee immune system. When we did have her test the extracts, she could not find a robust effect. There wasn't anything really going on that she could find. Antiviral compounds should work really well in cell culture, and yet that didn't seem to be what we see. So even though there could be antiviral compounds in there, we think that a lot of the effect is coming from nutrition and the regulation of the immune system and not antiviral compounds. Another thing that made us think that, we had a collaboration with Dave Hoekster's lab, and this was Alexander Dombrowski's senior thesis at the University of Buffalo. They went out and actually found their own Amadou mushroom trees, and they made their own extract under my directions. And they went ahead and tested their bees. They did not have a lot of viruses in their hives, but every hive that they had there outside of Buffalo had a gut parasite, Lotmeria passum. And the extracts, the hives that were fed the extracts, were able to reduce the level of this gut parasite. Now, clearly, antiviral compounds are not going to inhibit a flagellated protozoan gut parasite. So this is another thing. Very few compounds are going to affect both viruses and gut parasites of this kind. So again, all of this working together is saying that there's probably some really interesting chemicals in there. They're probably doing some really interesting things. But more than that, there's nutritional and immune modulating things that are going on, probably more so than the direct action against pathogens. So we're moving ahead with the FDA with this being a honeybee feed additive as opposed to an antiviral drug for bees. This will probably speed the approval in many ways because drugs have more hurdles you have to go over, more characterization of what exactly is in it. And well, as I said, the evidence to me is pointing that it's closer to food than it is to drug, although in some ways it's a spectrum. You know, some foods are quite healthy for you and contain weird drug-like chemicals. All right, so to kind of summarize this first part with the fungal extracts, bees really need a diversity of nutrients from their floral or sometimes non-floral resources. And I will say that it's likely that no single chemical or one single nutrient is going to be a magic bullet to fix bee health. You know, finding out that bees do better with sterols in their diet, that's going to be part of it. But we really need a bigger, you know, a broader view of their nutrition. Bees like that diversity of forage, and we should do our best to provide it. Wood fermenting fungi can provide many of these nutrients that bees lack, and it is certainly better than sugar syrup feed on its own. And then improved nutrition can help bees control microbial diseases. That seems to be where things are pointing at this point in time. So with that, I've noticed there's been some activity in chat. So let's see if I'll take any questions if anybody want to add those. Oh, I'm having trouble opening chat and I don't know why. Well, are there any questions somebody would like to?
I have a question.
Yeah, go ahead.
Hey, so a lot of us will plant certain flowers and things in our bee yards to give our bees something to eat. And I'm curious if we could get a hold of the spores of the mushrooms you use to impregnate a spore log that we keep in our apiaries to offer that up for the bees.
Yeah, Fungi Perfecti, our collaborators, they offer these medicinal mushroom grow kits so you can grow them and make herbal teas out of them. So in particular, I mentioned these reishi, or also sometimes called Lingzi mushrooms. You want to look for the genus Ganoderma. That's the ones that's really perform the best. The Oyster mushroom that Fungi Perfecti is growing, I don't know if they have it for sale for humans because the mushrooms are so hard and inedible that not a lot is done with it. But the Ganodermas have really worked great. And when you order a little grow kit from FP, it is the same strains that we are using. So it will be fairly easy for you to get the same materials that we end up using. Their little grow kits are not too expensive. So yeah, that's the easiest way to get that.
If you stop sharing your video, you may be able to get to the chat more easily if you're on a single screen.
Okay, there we go. Yeah, bee sourdough bread, right? Combination, a little bit of acidic fermentation that they do with their bee bread. That's kind of funny. I hadn't thought about that. I'll leave it to the Bay Area to remind me that, yeah, the bee bread's actually bee sourdough bread. Let's see, our previous speaker said fungicides sprayed in Napa County are a problem for hives he manages. Yes, I was involved in, there's a Washington beekeeper that coming out of the almonds had a very weird set of issues. And so I was the one that did the kind of the autopsy on the hive. I'm still not entirely sure what happened. We have samples in the freezer that I hope to process one day. We were able to use spray records to trace it to fungicide, of his different yards. The bees that had this weird thing going on, we traced it to a fungicide spray that seemed to be very much involved in it. However, when I pulled out brood from these hives, they were, the only other time I have seen things like this is with black queen cell virus. And black queen cell virus is typically asymptomatic in typical worker pupae and brood and even adult workers. And this looked like the bees were getting black queen cell virus in the worker brood. And so, I mentioned that, pesticides and pathogens and nutrition all interact together. If I had to guess, a fungicide nailed the bees' health bad enough that virus moved in and killed off all of the brood in a way that really took down the colonies. It was really sad to see full frames of brood, most of which were dead as a pupae. So yeah, fungicides definitely have problems, and the Bee Informed Partnership and USDA National Survey, they have found fungicides when they do the comb sample to find out what pesticides are in your hive. Fungicides came up at higher concentrations than varroicides, which I find remarkable. I think we put things directly in the beehive. That would be the chemical that's the most common, but no, these fungicide sprays that the commercial colonies encounter so much are actually in the comb at higher concentrations. So yeah, fungicides kind of safe for bees, but it depends on the dose and it depends on the interactions. Somebody said also fermentations breaks down the shell of the pollen granule, which makes the pollen digestible. Yeah, pollens have a very tough outer shell, and that acidic fermentation helps pop it. If you ever have time to dissect a nurse bee, they have an interesting thing where they digest most of the nutrients out of the pollen, but that pollen shell is really hard to get through. So they turn their hind gut, basically their colon, their large intestine, into a big fermentation chamber of pollen. So their hindgut's just swollen with pollen that they're doing this extra bacterial fermentation to try to get that last of the nutrients out of the pollen before they poop it away.
Wow.
Could you mix a Ganoderma reishi extract with powdered sugar and use it to feed your bees? Yes, the capsules tend to be fairly expensive. We haven't tried this ourselves because the liquid's been working so well. And, bees just like to process sugar spills, the sugar syrup, they process it and get rid of it. And so it's very easy for us to track consumption in cages with these liquids. With the powders and so on, it's a little harder about, it's harder to do exact science with the powder. It almost assuredly works, but we haven't gotten there yet. Some people do the dried drivert powdered sugar in the winter to feed bees. It could definitely be mixed in with that, but we haven't done any tests with the powdered formulations. Somebody says more about Metarhizium. That's coming up here in a minute. Also, can you please reshare the title of the paper? I wrote this title. I should be able to know it off the top of my head. I think it's Polypore Mushroom Extracts Reduce Honeybee Viruses. I'll tell you what, let me get this last one. Growers also spray insect growth regulators. I was very skeptical of insect growth regulators when I first saw the California almond spray records. But Reed Johnson's lab at Ohio State's done some good work to show that they're not as bad as you might think. Some of them, I say, still need to be tested more. And we do see weird pupil death things from IGRs. But in terms of what is sprayed and what the bees encounter in the almonds and how it affects their health, they're not as bad as I would have thought. But they could definitely be another one of those contributors and interactors to help bring down the colony. All right, I'm gonna go back to share screen and let me click back up here to, here we go, extracts of polypore mushroom mycelium reduced viruses in honeybees. When I usually just Google Stamets Naeger, and that's what comes up because it's our main paper together. Oh, that's interesting. I don't know which slide you are seeing now. There's the one with the title again.
I think the paper's link is on Stamets' website if you go there.
Perfect. Yes. In fact, you can even do a Google image for mushroom and honeybees or fungi honeybees. And I've seen this scientific graphic come up before, and then you can link to it that way. It's not too hard to find. Like I said, we intentionally made it open access so people can find it and beekeepers can use it. Nobody in the WSU program is a big fan of the paywalls they put behind scientific papers. And we've been making a very deliberate effort in recent years to, especially for the higher profile papers that beekeepers would be interested in reading, to make sure that they go somewhere where the public can access it without having to pay $50 an article, or some of these are really far too expensive, more than they should be.
Hey, Nick.
Okay.
Yes. Oh, no, I'm sorry. I didn't need to. Oh,
No,
I was going to say, I put a picture, I don't know if the picture will open or not, but one of my sort of my mushroom experiments with oyster mushrooms, I wanted to see if I could grow them inside the hive. And I configured a quilt box. It was just basically a medium super that had a queen excluder, plastic queen excluder underneath it, which I usually had filled with straw or burlap or what have you. And I emptied that out and I filled it up with aspen, shredded aspen chips, and inoculated it with some oyster mushroom spawn that I raised, and just to see if it was feasible to even grow it in a hive. And I managed to actually grow it in the hive, but whether or not the bees were using it or not, I couldn't really tell. But at least it was a fun experiment to see that you can actually grow, adapt a hive to grow mushrooms sort of in or on top of the hive. It was kind of fun to give it a shot. And I don't know if people want to try it out or not. It's kind of neat.
Yeah, I actually had some trouble getting fungus to stay alive in beehives in the summer. It was drier than I thought it would be, but this time of the year it will be good.
Yeah, periodically I'd bring out a hand sprayer and just spritz it with some water to keep it moist, and it was fairly enclosed. It was like a lid on it with just some vent holes around the side, and it not knowing if the bees were interested in it or not, but I did manage to harvest some oyster mushrooms out of it, and so that was kind of fun. But
yeah, it's really fun.
Interesting. That's why I want to see if it would, possible. I mean, the idea was that with the queen excluder being in contact with the actual inner workings of the hive, that the bees would, work the mycelium through the slits in the queen excluder as they were growing. I mean, that was at least the idea that I had in mind. But
yeah, I love oyster mushrooms. There's a fun thing that they can control nematodes. Oyster mushrooms literally grow a little lasso, and if a worm crawls through it, they'll tighten it and then digest the worm. So really fun. Yeah, it's a fun group to learn about. All right, I'm going to move forward with the second part here, which is using Metarhizium fungus to kill varroa. So, previously we've talked about mushroom extracts. Everything is put in the hive is dead and extracted. But here is our work with living fungus, and particularly trying to develop a strain of pathogen that will kill the mites. So varroa mites, I would say public enemy number one. Pesticides aren't great, the viruses aren't great, but if I had one thing I could just, snap off the face of the planet, this would be them. I really dislike these things, and they seem to serve no ecological purpose. Even mosquitoes, as larvae, they fit into an ecology where they do things in streams and so on. These are just a pain. I don't know what good they are. And anyway, like most beekeepers, I would like them to see more of them dead. So varroa mites primarily feed on the fat bodies of bees. This has been one of these revelations in recent years. We used to think they fed on hemolymph, honeybee blood, but we now know what they do is they like to dig in and scrape at the fat body that lines the exoskeleton of the bee. This is really tough on the bee because the fat body is it's really the primary center of metabolism, detoxification of pesticides and plant compounds, and immune function in general. And so, it's bad enough if a parasite was feeding on your blood. If it's taking chunks out of your liver, well, you're going to have trouble recovering from that. So varroa, they weaken bees, they're going to suppress the immune system, then they spread microbial pathogens, including symbiotic viruses. It's now very clear that there are certain strains of DWV and other viruses that have a tight symbiosis with varroa mites, where they make each other worse and they help each other spread. Overall, we get shortened lifespan. In bad enough cases, you get dead brood. And 20 years ago when I first started working with bees, it was so rare to find a colony. You have a sticky card with 4,000 mites on it, and you wouldn't see dead worker brood. And you could treat with Coumaphos, and the bees would still come through the winter. It really seems worse now. The number of mites needed to take down your colony is going down. And make sure you monitor your mites and get them under control, because they really are the top predictor that your hive will die in the coming months. Pathogenic fungi are quite common in nature. We as humans with our hot bodies, fungi typically don't like heat. They do a lot better on invertebrates or cold-blooded animals. But there are many, many different kinds of pathogenic fungi that go after arthropods. Here are just many, kind of charismatic pictures. Some grow mushrooms, some just grow little mold sorts of structures. And here's one most of us know, chalk brood. It's a fungal pathogen of honeybees. It does happen, the fungi are pathogens of invertebrates, fairly common. Well, about 25 years ago, I'll give you a little history of the story of how this came about. A beekeeper, if I remember, he's originally from Czech Republic or something like that. He approached at USDA and said that he was getting this mycar powder. It's an intimate pathogenic fungus. He was getting it from Europe and bringing in, and he claimed he could control varroa mites with it. Well, this got the USDA interested, and Rosalind James and Lambert Kanga decided to pick up the work and see if there's anything to it. There's some other work trickling in from Europe suggesting that there are some pathogenic fungi that could help control varroa, and perhaps importantly, the bees seemed relatively resistant and healthy when treated with it. Well, they started using commercially available strains. They did a lot with Metarhizium fungus, which is the one now that we are using. Metarhizium is well, it's extremely common, and there are many strains out there. There are ones out there for termites. Some are used for cockroaches. It's not incredibly common because it can be fairly expensive. And so compared to traditional insecticides, it doesn't have a widespread use, but it is extremely safe for humans and other mammals and so on. And so they did some experiments with Metarhizium. They did trials in Florida and then also Logan, Utah. It was somewhat promising, but they had very inconsistent results. One of the things that Rosalind wrote about was that the strains that they got could be extremely variable. And so you get Metarhizium strain for termites from company A, and it seems to work decent. And then you get Metarhizium strain from company B, and it doesn't do anything. And then furthermore, when they tried to grow it, they had these issues with contaminants and other problems like that. And then there's this whole question of how to deliver it to the hive. I mean, this was very early stages. They were literally putting spores in salt shakers and like shaking it over the hive, or putting it into a liquid suspension and just like doing pump sprayers on the hive. Anyway, they had some decent results. Similarly, William Meikle, USDA in Arizona, tested the Beauveria fungus against varroa, got some decent results, but here is where it was really unfortunate. He also had problems with consistency, and then there's a contaminant issue that eventually led to some queen deaths. And so, you know, some worker death, oh, that's okay. Queen death, that's pretty unacceptable for a miticide. The big problem that they had, and this is kind of coming secondhand from Eric Musson, was that there was a Pseudomonas bacterial contaminant in what they treated the hives with. And so when they went to do their big field study to prove that it worked, they killed a lot of bees because the company that they hired to grow it messed up. So it was very unfortunate, and that kind of led the higher ups of the USDA to be like, you've tried it, you got some results, but this is too finicky. And when we gave you the opportunity for the big field trial, you just killed bees. We're moving on. Well, about 5 years ago, Steve Shepard and Paul Stamets, or I mentioned Paul had called up Steve to discuss bees foraging on his mushroom beds. They started this collaboration using fungal extracts to improve honeybee health. However, they were very aware of the other fungal bee things that had happened in the past. And so they wrote a grant to revive the work with fungi to control varroa. They got this funded by the Whittier Foundation, and that led to the hiring of younger me and Dr. Jennifer Hahn, plant biologist, mycologist, geneticist, and now a bee researcher also, to develop a fungal strain and really see how far we could push this fungal control of varroa. Little side note, Jennifer and I are now married. So the fungus, Metarhizium. We really like this one. You know, again, Beauveria, we thought it was a contaminant, but the idea that there could be bee death, we didn't like that. So we focused on Metarhizium as our primary fungus. And again, no signs of bee mortality. It seemed the safest one for bees, which was paramount. You know, above all, do no harm, right? Second thing, it is extremely common in US soils. So when Jennifer was teaching the graduate level mycology class here, she went to the community garden down the street and dug up some soil and cultured fungi out of it to see what was in the soil. Metarhizium was the second most common fungus in that sample. It's really common in US soils. It's all over the US. And you know, it's down there doing things like waiting for a little worm or a cricket or, you know, some ground dweller to crawl by before it infects it. After it consumes all of the body, the mycelium then grow back to the outside where they produce these greenish or sometimes brownish spores. It is EPA approved. Here's Bioterminator, Metarhizium. Looks like here they might be trying to kill termites. Met52 was a patented strain that even Monsanto mass produced for a while. These are very safe for human contact, very safe for human consumption, and again, safe for bees. So this was where we decided to kind of start the research because it seemed good for bees, good for humans, and it's native. We're not introducing anything new. Starting out, we were doing some really complicated science, taking a petri dish of fungus and plopping it upside down on the top bars of the hive. We would put a plate in the top here and then between the boxes also, you know, in the brood nest area, and we let the spores kind of rain down on the bees for a period of time. The bees actually don't like having living fungus in the hive, and they scrape the petri dishes clean. The agar is left behind fairly clean after just a day or two of the bees working it. We did an interesting thing early on. This fungus can produce it can produce pesticides, basically. When it gets into an animal, it can begin to produce these chemicals to help overwhelm the immune system and kill the animal so it can consume all the little tissues that are in it. Well, we got a strain that delays producing spores, and we treated hives with that too, to try to find if there's any chemical effect versus the spores landing on and like eating the mites alive. Well, basically what we found there was that it eats mites alive. It's not the chemicals that are inherent within the fungus. Well, here's what we see. When I'm in laser pointer mode, it does not want to let me play the movie. All right, so here are two mites that we put in the freezer briefly to kill. We dipped them in spores and then put them under the microscope camera and did a time lapse. So here are the mites. They get a little bit of rigor mortis, so they're still moving a little bit. The spores are now kind of creating a, they almost grow like a root that like digs into the mite, and then it consumes the tissue on the inside, and eventually, like we saw in the earlier pictures with the cockroach, the mycelium kind of pushes back out and they grow all of these greenish brown spores. Oops. Okay, so there we go. What we decided to do was start with repeated selection of strains. So we would grow the fungus up, treat a colony with it. The colony would have sticky cards, so we could collect the dead mites. Then we would pull out the sticky cards, count how many mites had died. We would do an ethanol wash to surface sterilize, and then we would plate them onto Petri dishes. So on each of these Petri dishes, there are 6 mites in the middle, and on this one, 5 of the 6 grew fungus out of it. Here, only 5 of the 6, sorry, 6 of the 6 grew fungus out of it here, 5 of the 6 here, and only 1 here. So what we're looking for is fungal lines that kill the most mites per dose, and we also want to kill mites fast. I don't want to get too much into the ecology of wild fungi and animals, but they tend to kill slowly. So they produce spores over a longer period of time and so on. We want fast killing with varroa. So we select for the deadliest and the quickest killers, and we repeat the cycle, just repetitive selection. This fungus is completely asexual. So there's no real breeding. We can't cross lines, but we can repeatedly select for the better subgroups that come out of each line. Well, like a lot of fungi, Metarhizium spores do not like heat. I mentioned this is a soil-dwelling fungus typically, where it's nice and dark and cool and moist. And beehives are not cool. They're really quite warm. Beehive temperatures will kill spores after a few days. And so even Rosalind James back in 2009 in one of her final papers before they gave up showed that when you store these at 4 degrees, basically store in the refrigerator, the number of spores that germinate is fairly decently. However, if you store it at 35°C, a beehive temperature, you get this rapid die-off of spore viability. You can extend it out a little bit by giving them an oil formulation that kind of helps prevent the spores from desiccating in the heat. But overall, heat is killing the spores. And I'll show you some more slides here. These were some of the strains that we work with, one of our starting strains. So spores stored at refrigerator temperature and then germinated at room temperature or beehive temperature. So even when they're stored at a nice cool 4 degrees and they should be healthy, when you germinate them at room temperature, they grow these nice long, it's called an aprosorium organ, but this really is almost like a plant root digging into the ground, except it uses it to dig into the mite to get to those internal tissues. It's a fairly sophisticated chemical drill to get through the exoskeleton of the mite and to the inside. Well, at beehive temperatures, these are very much stunted. You know, we have some spores that don't grow at all. Most are alive, but they're not super healthy looking. They're just not happy at that temperature. If we store the spores at 35 degrees, they are really not happy. Most of them die. When we bring it back down to 25 to germinate it, some of them survived. We get an okay germination tube, but germinate at beehive temperature, that's really a bad combo. They do not do well at beehive temperatures. So Jennifer and I sat down, we read a lot of scientific papers, and we devised new techniques to basically give this fungus a kick in the butt and make it evolve to like heat better than it did. This technique is called directed evolution, but really we're just using incubators in the lab and some chemicals and some other treatments to help push it, to evolve it in the direction that we want it to. We had these different treatments. One was a combination of oxidative and osmotic stress, and that included some mild mutagenicity. So we're trying to really kind of stress this fungus out and make it evolve. And then second, there's some great things in the literature showing that if you starved the fungus of nutrients, this makes it evolve faster too. You know, you put these things under stress, they have to change to stay alive. And so all their genetic machinery and so on kind of changes in a way that you can kind of push them to evolve a little faster. So we designed this technique. We had these alternation of generations to maintain lines, but everything went through both of these treatments several times. We then set up a stationary apiary, that's what we call this experiment, for repetitive field selection after our directed evolution in the lab. We call it a stationary apiary, which is almost a euphemism for saying, hey, Steve and Brandon, please don't take my hives to the almonds. You know, it's too precious. We're doing long-term research here. Leave them where they are. So anyway, we set up this 48 colony apiary, and half of the bees got Metarhizium treatment and the other half did not. The ones that got Metarhizium treatment, we used for continuous selection for better strains to kill more mites. This was a huge project. This really was a couple years of my life. Perhaps it's not surprising Jennifer and I are married because we had a lot of 15-hour days in the lab, counting mites off of sticky cards, sterilizing the surface of a mite and getting them onto Petri dishes. Over 20,000 mites were screened for fungal growth. I don't know how many Petri dishes we went through. Actually, 20,000 divided by 6 is about how many we went through. It was a monstrous amount of work, but we did get some results. So here's what we got in the year following our incubator selection. Now, when you mutagenize and stress out a fungus to make it evolve, imagine this. Most of those mutations are not healthy for it. The fungus that came out the other end was a little decrepit in a few ways. It wasn't that great. And so our first treatment, this is the percentage of mites that died as a result of fungus over the experiment. These are continuous sticky cards. So we collect every mite that dies in that hive and do our little autopsy to see if fungus killed it. Well, generation 1 treated here, we get a little blip where it comes up to maybe 5% of the mites die, and then it kind of goes back down. That was quite disappointing. But we took the fungus that killed mites and we recultured it and made a second generation, and boom, we're up to 50%. So that was immediately good. We're back to where we started anyway, but this fungus should be surviving better in the heat. So let's give it another round of selection. 3. Okay, well, we didn't get much higher here, but the tail of it's kind of drawing out. The fungus is beginning to kill mites for more than 4 to 7 days. It's beginning to push out more to 7 to 11 days. And then we finally made our big selection event where almost all the hives were treated with a good thermotolerant strain now that we selected for 4 generations, and we got that. So we're killing many, many more mites. The window of duration is much longer. So you can put one treatment on and have it affect the mites for, instead of 4 days, we're now looking at a month to a month and a half. If you think about total mites killed, it's like the area under the curve. Each treatment is now killing many more mites. And at this point, we are still just taking a Petri dish and turning it upside down on the top bars of the hive. Nothing more sophisticated than that. This is something kind of fun for those of you that might be into fungi. Here is a spore print of our Petri dish on the top bars of the hive. You can see the greenish colored spores from Metarhizium. We went through and swabbed this in the early spring in March. Our last treatment had been done in September, and we were able to recover living spores that survived 6 months over the winter. Now the hive isn't super hot during the winter, but still they survived, and that feels like such a major victory. When we look at these 48 colonies and what happened to them, well, perhaps not surprising, those that did not get a mite treatment began to die off. And here's where I wish I had things to do over again, because when you get here where half your colonies are dying from mites, the mite bomb effect is real, and it's so dramatic. Our colonies that were receiving treatments got absolutely overwhelmed with mites pouring in from the other colonies. We could not control it, and the whole yard died very quickly. So things looked really good until everything died. But I think it's still very promising that we were able to extend the lifespan of the hive until everything went to crap. We also had these problems of there were yellow jacket attacks coming from one direction, and it was absolutely nailing our treatment colonies worse than our control colonies because of the geography that we had them set up. It was really unfortunate that we couldn't control the situation better, and now we compare our treatments rather than comparing it to no treatment. We typically compare it to something like oxalic acid. That is another decent treatment that's out there. All right, well, at the end of all that, let's do a little more during the winter to see what happened with our strain. So spores germinated at beehive temperatures, 35 degrees C. This is our original strain before we did the thermotolerance and the directed evolution. And again, as we see at 35 degrees C, well, most of the spores germinate. There's a couple dead ones, but they're there. They're not looking super healthy, but they're there. This is our post-selection strain. The spores just look a lot healthier. The germination tube is significantly longer. And as we showed, they can infect and kill more mites. So this was kind of our proof that we truly have created a new strain of some kind. It's much more happier at beehive temperatures, and it does kill more mites. So we feel like we have the strain going pretty well. We're continuing selection. Every time we do an experiment, we're keeping the original line, but we're also continuing to see if we can push it to get a little bit better, a little bit better, and a little bit better. But at the same time, things are now so promising that we're now looking at production methods. This was really nice in having a collaboration with Fungi Perfecti. I had grow masters to talk to, I have my, volumes of scientific literature to see what people have done before, but I was able to talk to some of the growers at Fungi Perfecti and just say, oh, well, when you work with rice, do you have problems where it gets too mushy or too wet or, that sort of thing. And so it really helped kind of jumpstart us to a pretty good place. Grains are a pretty good substrate, particularly smaller grains, corn gets a little big, but some of these smaller grains that might be about the size of an insect work really well for this fungus. We're currently trying to identify the best mix of grains to make the fungus happy. We've already learned some things like here is whole barley and then pearled barley. You can see there's a lot more fungal growth on the pearled barley. Even though this fungus does really well at drilling through a mite exoskeleton, it can't get through a barley hole. So if we de-hole the barley, pearled barley, the fungal growth is a lot faster and a lot better. So we're learning about that and hopefully making some headway. Here is a treatment we did. We've actually done a couple of these now. Here's a treatment we applied 3x over 3 weeks. And the hives got either an oxalic acid drip. It was 3.2%, might have just been 3%. And then the 10 mils between each frame dripped across. Or the bees got these little bags. 120 grams of colonized rice. I would contest that this is not a big treatment. We did go ahead and put a spacer on, so the lid, just so there was room, and the bees had room to work the fungus and not get, squished by the lid. Yeah, they still have a feeder in here, but we didn't use that. These bags are made out of a banana fiber. They're actually kind of weak bags, but we chose this on purpose because the bees can chew through it really easily and begin to work the grain. I mentioned that bees don't like fungus in their hive and they begin to clean it out. So when we put this in, the bees chew holes in the bag and one group of bees begins to pull the grain out and drop it to the floor. And then other bees proceed to drag it off the bottom board and drag it out of the hive. While these bees crawling all around and working the fungus, this helps spread the spores around. When the bees do this, the spores are getting into all those hairs. And then if a mite crawls through those hairs, it gets infected. So we think that is really good that the bees do clean up this fungus. And so we try to go with a formulation where the bees can clean it up. And hopefully at the end, the beekeeper has no old plastic strip or anything like that to pull out. The bees just chew it all up. And whatever's left in there will not be harmful, biodegradable, organic, and all of that. One study we have analyzed completely, Metarhizium did create a steady mite fall. The oxalic acid would create a very early spike. We'd see immediate mite death, but then it would go back down. The Metarhizium created a much more steady, constant mite fall. The Metarhizium treatments held mite steady, but the oxalic acid actually increased in mite levels. So we outperformed oxalic acid in this study. And I will point out that June in Pullman is hot, but it worked anyway. So that's great. Sometimes these fungi are discussed as cool weather treatments because they only work, they generally don't like the high temperatures. So this is extremely promising that we got it to work, and we got it to work in June during the hottest month, and we outperformed oxalic acid. I just had a meeting with a grad student at Brandon Hopkins that was doing an indoor wintering experiment. And she wanted to use Metarhizium as one of the treatments after indoor wintering to control mites. And again, it looks like we performed in that study on par with oxalic acid. We didn't outperform it, but still giving beekeepers another non-chemical, very safe for bees treatment. I'm very pleased with how things have progressed. We're doing a few other experiments. Here's some felt strips impregnated with Metarhizium. We did some work to find the right polyester fabric that can be autoclaved and withstand autoclave temperatures so we can do the sterile growth of the fungus. Here's different fungal strips I created that we strung on a wire and then put down into the hive. This seems to work quite well too. It would be very easy to treat your hives with strips you put down in the brood box. But this one is tricky production-wise. I haven't abandoned it yet, but the banana fiber bags with the fungus growing on grain seems to be working quite well. And I think we're gonna move forward with that. Maybe try to make a rice cake sort of a thing for the bees. Put it across the top bars. A couple other fun things to finish this up. We have a really talented undergraduate researcher, Riley Schultz. She's a biochemistry major. She was pre-med, but has fallen in love with bees and bee research so much that she's now off to Purdue to work with bee health there. She's done qPCR to study the effects of Metarhizium on the bee immune system. So this is an interesting idea that somebody else posited in the literature previously, that if you give bees a fungus that is not pathogenic to them, the immune system can still recognize it as a fungus and boost itself in a way that can protect the bees against other microbes. Well, first thing we found is, I should back up. We modified our extract cages so the bees could get Metarhizium in the cages. Here's a control cage. Here's one with a tiny strip, a low dose, medium-sized strip, and a bigger strip here. We grow the fungus on the strip in the way I showed you on the previous slide. And then this is interesting, the bees clean it off. And so again, the bees don't really like it. They scrape at it with their feet and mandibles, and you get dust all over the floor. But as they work it, they spread it around themselves. Excuse me. Well, one thing that was really great to see, and I wasn't really expecting this, there were mites on bees in the cages, as is inevitable when you take bees out of a hive. There were mites with them. But the bees that got Metarhizium treatment did live longer. So that's an immediate thing. That's what we want to see, is that we are helping bees live longer. We did see mite fall as a result. It's very hard to pull mites out of these cages. We didn't collect great data. I think we did have significantly more mite fall in the cages that got Metarhizium treatment, but our data is not good enough for me to really say that. But we did get longevity extension in ways that it has to be related to killing mites, right? I would think so, but I can't say for certain that we killed more mites in those cages. But Riley's main thing was she wanted to look at the bee's immune system. So here's one of the highlights we have so far. This is okay, let me go back. One more. Here we go. This is a phenol oxidase gene. This is a very interesting gene. It's involved in melanization and encapsulation of foreign invaders. So bees, they have a bacteria, a fungus, even like a tiny parasitoid wasp in their body, they will take and cover it in melanin, almost like an oyster makes a pearl to protect itself. The bees will cover it in melanin so that microbe can't do anything. It suffocates. It certainly can't eat or grow or divide because it's just balled up in this melanin ball. It's also very important for wound healing. And they can also do things like melanize potential pathogens. So certain bacteria or nosema in the gut, they can melanize those to help get rid of them. Well, we found with the highest dose of our Metarhizium over here that all 3 collections we did over the course of a couple weeks, the bees appeared to have a boosted immune system in this one respect anyway. Not all aspects of the bee immune system were boosted, but this is one where we did get a significant result. Another interesting thing, I mentioned that many fungi produce oxalic acid naturally. And if something produces a chemical, it's probably resistant to it. It would be very strange for a fungus to produce oxalic acid if it died from oxalic acid. Well, here's an interesting thing. This was a little collaboration with an Australian lab, but they looked at Metarhizium under an electron microscope and found that there are these crystals of oxalic acid, and oxalic acid makes some very obvious crystal shapes. It looks like Metarhizium does produce oxalic acid, and so that's an interesting thing that, well, maybe there could be some synergy. So we had a master student. She's originally from Turkey, Fatime Yedis. She was very interested in biocontrol. And so what she did was she ran a series of carefully controlled laboratory tests using a system called a Potter's spray tower. It's a device that sprays a very specific dose of something onto something. It's used for pesticide studies. And so what we did is we had her make different suspensions and concentrations of oxalic acid with or without Metarhizium spores and just see the effect on mites. Basically here the y-axis is percentage of mites that were killed. In control, it's down here. 1.5% oxalic acid gave us a midpoint of control. When we do science, frequently we want to give something the opportunity to control better or control worse. So 1.5% oxalic acid was our intermediate dose that kind of killed half the mites. Then what Fatime did was she took a very low dose of oxalic acid that doesn't kill many mites at all and begins to add spores to it. So 0.5% oxalic acid, really like less than 20% of what you typically treat a hive with, doesn't kill too many mites. But if you begin to put spores in it, well, it's a little bit better than the control. You put more spores in it, well, it's better, but there's wide variation. And as you add more and more spore concentration to it, you get to the point that it is controlling things better than oxalic acid, even at the higher concentration. So this was a great proof of concept that these two treatments can synergize, they can go together. We can even mix it up into the same liquid solution, and the Metarhizium spores are perfectly happy in that oxalic acid solution. So we haven't done a field test yet with this 1-2 punch together, but this is another great way that if beekeepers want to do an organic treatment and avoid amitraz and some of the harder chemicals, it might be a great combination here. All right, a lot of thanks to the WSUB team. Dr. Jennifer Hahn really is, apart from myself, we're the most responsible for this work. We've spent so many hours making this happen. Steve Shepard kind of runs our WSUB program. I have great respect for Steve. When he started at WSU, the bee program was 3 hives and some space in a garage. Now we've just opened a gigantic new facility in Othello. We have 3 trucks. It's really quite a big lab. It's now Steve, Professor Brandon Hopkins, and myself, 3 professors in this program. We've gone from 3 colonies to about 400, and really things are taking off. And it kind of all started with Steve. Lori Karras was a mycologist. She just retired, but she was really instrumental early on in teaching Jennifer and myself how to deal with fungi. Is also very interesting that MET52 strain that was really popular. I said even Monsanto was manufacturing it for a while. She found the great-grandparent to that back in the 1980s during her graduate career. So it's kind of interesting that her long career in mycology was bookended with this work with Metarhizium. And then the WSUB team's really fantastic. Working with bees and working in the almonds is not easy. I don't know if you remember all that rain a couple years ago, the mud fest that resulted. But yeah, especially with the extract experiments, there were many times when I had the crew out there from sun up to one day we worked until midnight. And I don't know if you've ever done beekeeping by headlamp, but don't. It's awful. But I'm so thankful that I'm with a group that stuck with us through it. Oh yeah, and one more of us getting the truck stuck in the mud again. That poor rental car, when we returned it, there must have been like 10 pounds of mud in it. Anyway, thanks to the bee team. And then also our funding resources. Is kind of interesting that unlike most academic science, we've been really crowdfunded. We've been funded largely by donations. Initially, Steve and Paul got the grant from the Whittier Foundation, but this was really ultimately the support of the Rhodes family. They provided the initial grant that hired Jennifer and me to get things going. We've also parlayed that into grants from the Washington State Department of Agriculture to help pay our salaries and so on as we do this work. Paul has been a financial supporter as well as just a champion of bee research. And Fungi Perfecti, not only have they given cash donations to help run the lab, but their in-kind contributions over the years have been extremely significant. Barrett Foundation, Jonathan and Kathleen Altman Foundation, California State Beekeepers, and then also numerous small donors. And so if any of you do have interest in helping out, you can go to bees.wsu.edu. And when you scroll down to the bottom, there's a little button for Give Now, and that takes you to the WSU Foundation that handles charitable donations. And if you link through the bees.wsu site, it should autofill honeybee and fungi mushroom research. So that's all I have. Thanks. I talked for quite a while, but hopefully everybody enjoyed that. And I'm going to stop my slide share. I'll take any live questions. Oh, sorry. Okay. I'll take live questions, and I'll also check the chat here in a minute.
Hi, my name is Ko, and I started using reishi teas around 2003 on my bees because the bees told me to, and it's been very good. Washing the little teacups is a drag, but I started working with Metarhizium and Acephali, the MET52, and at the counterculture labs here. And I had the same problem that you did. So I really appreciate your methodology of doing the Petri dishes out of the varroa, but in my lab, because it's open source, we can't bring anything living into the lab. So I have to, this idea I had a long time ago is what your methodology was, and I have to figure out a way to do it on my own. But it's really exciting. It's just like, yes. Great work. And I, you know, you said how many dishes you had. I just had, you know, my first trial in the lab, you know, my first project, and I think I had about 30, and that was overwhelming anyway, you know.
Yeah. Yeah.
Thanks for your work. I'm so excited.
Yeah, and one of the things that I find particularly exciting about this work is, you know, beehive temperatures are hot. And as I mentioned, these fungal treatments are frequently referred to as cold weather treatments. Yeah. So as soon as we get bees squared away, we can begin to branch out and begin to try to develop strains that are good against fungi, or another of my most hated creatures on the planet, the two-spotted spider mite. Some things like that. We can begin to see if we can take other strains, maybe even the strain we already have, and begin to do these directional selections to really make fungi that are better than what we find in the wild, and you know, kind of push them to help us out. And it would really be great in the age of so many chemicals to have a few more biocontrol options.
Don't forget tropolalaps.
Oh, that I have nightmares about that mite.
Because if the Metarhizium would transfer to that, that would be good.
Oh, absolutely. I shudder to think of that mite arriving before we have varroa under better control than we do. For those of you that aren't aware of it, tropolalaps is another mite that can infect European honeybees. In places like Thailand, it has host jumped in a story that feels very similar to what varroa did. And it reproduces faster. The mites run around faster. They just seem even more problematic than varroa. And the thought of getting another mite in this country is just, let's hope it doesn't happen.
You just made me think of something. Maybe that's why the giant Asian wasp is trying to get out of Japan.
Yeah, sad to say, I'm pretty sure that was brought in by somebody growing them for food in the Vancouver area, and it just escaped. I find that so sad. That's going to be such a pain for beekeepers. That hive that they were able to collect, the hornet hive they were able to collect, it already had been creating reproductives. There's going to be more hornet hives out there. And yeah, it's kind of sad. It's because somebody would make some specialty food in Canada.
Well, if La Nina is cold, maybe they won't make it.
Well, in the areas that they live in, China and Japan, they'll be able to make it here.
Ah.
Yeah, I know that's not what anybody wanted to hear, but that's almost assured.
The other thing I wanted to point out is one thing, I don't know if you know about almond cultivation, but the fungicides are never given singularly because of the labor. And it's just like the cocktail that they've killed the HIV. It didn't work until they had a cocktail of drugs. So the cocktail of fungicides are much more lethal to the bees. And you probably know that, but you should say that too. You know, just part of it.
Yeah, when I lived in Illinois, we'd have these problems where the aerial sprayers in particular were not good about cleaning out their tanks between sprays. And when they were supposed to just be spraying fungicide, there were absolutely pyrethroid residues in there in a way that could zing our bees. Yeah. So, yeah. And there's also issues where there's so much with chemical interactions. The adjuvants that make the chemical soluble or, you know, help it spread on the leaf, those can make the chemicals more toxic to bees. There's still a lot we're learning in terms of the interactions. All right, I had somebody ask, what's the name of the product from the Stamets site? So tell you what, here, I'm going to share screen this way. Hopefully you can see my browser. Here's one of the Ganoderma mushrooms. I just Googled Fungi Perfecti Ganoderma. Here's a Ganoderma lucidum culture. This one's fairly expensive. Come down here, reishi plug spawn. So if you want to grow your own reishi logs, out of stock, but quite a bit cheaper. And let's see, this one looks like a Resinaceum picture. And so I'm guessing this is, oh no, this is Ganoderma lucidum also. So they're definitely there, yes, out of stock, but not too easy to find. There are other people that sell these other than Fungi Perfecti, but
in our area, Far West Fungi in what's that? Moss Landing will sells those, and they sell them out of the ferry building.
Okay, yeah, it's becoming much more common. I have a sister that lives in Colorado Springs, and I'm kind of amazed how easy it is to find a reishi tea in a coffee shop there.
Mm-hmm.
Let's see, somebody asked, can mites become immune or resistant to the fungus? Possibly, but if that happens, we will keep evolving the fungus to hopefully stay one step ahead. You know, with this, with the way mites have evolved resistance to chemicals over and over, generally what we find with insects is that resistance to biological pathogens is slower to develop. And because we do get to continuously reselect and improve the strain, hopefully, if that does happen, we can stay ahead of it in some ways. One thing I haven't mentioned is we have found that even though we can do the autopsy on the mite and find that the fungus killed it, the fungus does not produce very many spores in the hive. Now, at first I thought this was kind of disappointing because I imagined treating a hive and you let a whole ecosystem develop and the dead mites produce spores and those can reinfect new mites. That does not seem to be happening. At first I was a little disappointed in that, but that actually gives us control in a way that is good. And what I mean is we were able to take something that was somewhat pathogenic to varroa and turn it into a good pathogen for varroa. There's that little bit of me that's afraid that some weird confluence of events could turn it into a bee pathogen. And if it was to like run wild within a hive endlessly, maybe it could evolve into a bee pathogen. I don't think that's likely, but now that we know that it's not really reproducing in the hive in that way, that means that we can keep our strain going, keep continuing to make sure that it kills only mites and not bees, and that way we have kind of control to make sure that we never develop unintended consequences. It's probably not going to get back into the soil and so on, and do anything unexpected on soil-dwelling mites. And so, yeah, you know, initially I thought it was kind of an unfortunate thing that we don't produce very many spores out of the dead mites. But I think it's one of those that is just ultimately safer for the bees in the end. And that's really priority one. Early on, I had somebody ask me, you know, how do you know you're not breeding a bee pathogen? And I said, well, I'm trying my hardest not to. And, you know, but now we have some good evidence that it's not even really possible. It's very unlikely that this is going to jump to bees given what we've seen in the hive. Somebody asked, any restriction on medicating when honey supers are on? So with Metarhizium, this is a hope that we can have the ability to treat when honey supers are on. With things like Hop Guard and so on, people are kind of using it when honey's on, even though they probably shouldn't be. This is one that, again, we'll go through the appropriate EPA channels to get it registered. We're hoping that it could be done because it is just so human safe. But we don't really know. That's a bit down the road when we begin to seek EPA registration. Somebody else has said must be stored in a cooler. Yes, if you want extended life. So if we do store it in a refrigerator, the lines that we have become nearly immortal, decades, and the spores themselves really do, they survive for a year or two. They do quite well in the cold. When you bring them up to room temperature, they get more like, they're like seeds. bring them up to room temperature, they begin to want to grow and do things, and that will shorten the lifespan. So you keep a lot of seeds, cooler and dry and so on. And these spores aren't too different. So, it would be the sort of thing we manufacture, we keep it cool, but then whenever, it's time to ship, well, it's fine in the mail. It'll be good. We might have to put in one of those silvery plastic sleeves to keep the UV light from the sun off of them. That could be a bigger problem for that. But yeah, it's fairly stable now. So there's a chain of cold will not be necessary for you to use this in your hives. Somebody asked, what about cardboard wallboard shims? Do you think those would work? Probably referring to strips. Yeah, that's interesting. There have been people that have grown Metarhizium on old egg crates, which I find interesting. Like, that fibrous pressed paper, like you might get a dozen eggs in. There are different things. Yeah, we haven't gone too far with that. We really are kind of pushing the grain right now because of the quantities that we can grow per. It's really a labor cost when it comes to the strips. It's hard to sterilize the strips and get them inoculated in any sort of volume. Whereas with the grains, we're already growing it in like 3 kilo bags, and I don't see much of a reason why we couldn't make it a lot bigger than that. So we're kind of heading with the grains first. But yeah, ideas for the strips, I still find interesting. I still like to tinker with that when I have time. And let's see, somebody asked melanization. Yeah, that bee immune system, it's very interesting. It is melanin, like what you have in your skin and so on, except the bees make it in a way that it polymerizes. So it's going to make a polymer, like imagine a glue hardening. There are cells that free float in the bee's body that when it finds a foreign invader, they begin to just glom on this melanin that polymerizes into this hard shell around the invader. It's a really fascinating system that insects and a few other invertebrates have. We don't have anything like that in our body, but we have antibodies instead, which might be better. Randy Oliver speculated that oxalic acid interferes with varroa's ability to smell the right state of the larvae to infect it. We have seen immediate mite death after treatment in a way to suggest that it's more of a direct toxicity. However, it would not surprise me if there's multiple modes of action. So yeah, it's certainly possible. And just because I say there's more of a direct toxicity, these are not mutually exclusive theories. It could definitely be interfering with varroa's ability to smell, or some of you might have seen when Randy was talking about the damage that oxalic acid can do to like the tarsi of mites. Their little feet are kind of fragile. And sometimes after oxalic acid treatments, their feet look kind of messed up. So maybe that's another part of it. We don't really know everything yet, but there's a lot of people working on it. Says, sadly, the almond pollination is a great place for mixing and redistributing bee pathogens across the country. Oh, how true that is. I taught honeybee biology this fall, and we talked about tracheal mites, and I say, oh, they came in 1985, and then they were everywhere. And then you talk about varroa mites came in around 86 or 87, and then they were everywhere. Yes, it's an epidemiological nightmare having 60% of hives from every state of the union shipped to one location to share diseases before going home to spread those diseases all over. It's really unfortunate in that way, but with the way American agriculture is structured, I don't see another way right now. We have to break monoculture before we can fix those issues. So we're just going to have to deal with an epidemiological nightmare until we make broader changes to the way we grow our food. Does a bioflora type of hive help support the growth of spores? No idea. I guess that's my answer there. I really don't know. I have no experience with the bioflora, so I'm really not sure. Can you place too much inoculated grain in the hive? Probably not. So I've of course been interested. Can I kill a bee with Metarhizium? And so a couple years ago, Jennifer and I took a dose for an entire hive, and we gave it to about 50 bees in the lab. And what happens in those cases is the bees are mostly okay, but what we find is they develop dysentery. What ends up happening is they get covered in spores, and then the bees, they do their little grooming thing, and they end up ingesting a lot of those spores. It doesn't appear to go pathogenic in them, but instead, I guess consuming all of this material disrupts the gut microbes. Maybe it messes things up in a way that nosema goes from latent to problematic. We're not really sure, but when I gave the bees, the triple mega dose, they pooped in the cage, and once that happened, they didn't live long after that. Once feces goes everywhere, the bees don't do well. So it didn't directly kill them, but with the mega doses, you can begin to affect the bees' health a little bit. I actually, again, though, that was a whole hive, that's a dose for 20,000 bees we put on to 50 bees. So I think you have to put at least a whole other box of grain, a whole super of grain on top to get anywhere near that dose. It'll be difficult to overdose the bees in that way. Since the mushrooms produce oxalic acid themselves, doesn't it seem to imply that this is just another mechanism for treatment with oxalic acid? Probably not. If that was the case, then some of our treatments with the ones that did not produce spores should have had some effect. And there's also things that the amount that's produced, that's difficult for me to fully refute because I can't really prove concentrations that they make. We know that the spore has to infect the mite, has to get into the internal tissues and begin to consume the mite to kill it. Is oxalic acid then the mechanism of killing, or a primary mechanism of killing, as opposed to consuming the tissues, producing these pesticides called destructins or other things? Can't say for sure. I think there's more going on than just oxalic acid, but I can't say for sure at this point. Is there a tropical strain of the fungus that will do the same thing at a higher temperature? Not that we know of, and not that's available in the US. Going to the tropics where these fungi are not uncommon would be very interesting. In the tropics, ants are fairly susceptible to some of these, and they can be a major control element in wild ant colonies. So there are definitely tropical fungi that will infect and kill insects in the tropics. That is assumably at the warmer temperatures. But yeah, there hasn't really been the background of somebody going out and looking for ones. And the other thing is we know very little about these, especially the diversity of fungi that kill insects. When we begin to talk about things that kill mites, that is so small and so easy to miss. We really do not know much about it. One day, again, when the bee problems are solved, I would love to do a broader biological screen, do more with the ecology of these fungi. I didn't even mention this, but this Metarhizium fungus can also form mycorrhizal associations with plants. So when it's in the soil, waiting for an invertebrate to crawl by to infect it, it can associate with a plant root and do a mycorrhizal plant fungus interaction where they trade water and nutrients and minerals and sugars back and forth. And so there's a lot more about the ecology of these things. I would love to study it, but for now, we're just trying to save the bees. Is there a hive treatment, milligrams of spores per hive, milligrams per hive, grams per hive? It's gonna be more like grams per hive. USDA, when they were isolating spores off of growth substrate, they did treat with up to 40 grams of spores per hive. We are doing more like 5. Is this control the mites? Will it also deal with the ants? Well, this is kind of interesting. Fungi Perfecti has kind of handed, they don't necessarily want to be in the pesticide game. And in terms of our work with Metarhizium, we've taken it we're just dealing with it on our own. We kind of loosely collaborate with Fungi Perfecti, exchanging information back and forth. However, we're doing the bee thing, but Paul has another little project trying to develop one for carpenter ants and maybe termites. He has some strains that seem to be more attractive to ants, where ants are usually fairly repulsed by fungus. And so they're still tinkering around with that. So yeah, I guess to answer the question, yeah, it will need to be a different strain, but there are people out there kind of tinkering with strains and trying to develop something for ants. Oh, Argentine ants, yeah, we don't have to deal with those here, but wow, what a nuisance. What an ecological nightmare for that matter. You know, ants are so important for the functioning of soil ecosystems, and everything grows out of those soil ecosystems. And to see the level of disturbance that importation has caused is really unfortunate. Yeah, so maybe so. Yeah, it's hard to know where all this could go. One of the things that's definitely come to mind, the fungus is still going to be relatively expensive to grow. As compared to a chemical pesticide, it's going to be relatively expensive. And so one of the low-hanging fruit things that seems to make sense, what is a high quality, expensive crop that is grown indoors and is susceptible to mites, for which people want a lot of organic production of it? Cannabis. So one of the things that might be a next step is to work with cannabis growers on mite protection in those sorts of greenhouse situations, because nobody likes to smoke pesticides. All right, that's all I see in the chat. I have a hand up from Tom, if you have a question. Okay.
That was me from earlier. I was asking about the plugs of fungus.
Yeah.
So
Okay.
Thanks for answering. That was great. Great talk.
All right. Anything else? I've kept you for 2 hours now. Thanks for hanging in there.
Thank you. Good stuff.
Yes, thank you very much.
It's been really fun to join the WSU lab. My PhD lab was very, I said we study bees like people might study fruit flies or mice or, so on. And yeah, I made some really fun little neuroscience discoveries about how drones control instinctive behavior and things like that. But it's been a lot of fun to join the WSU lab, and really, we like to say it's a boots on the ground sort of approach. We are going to go into the almonds. We're going to kind of deal with these beekeepers directly, try to identify the problems as closely as possible, and jump straight to a solution. And yeah, it's been really great working with this group, and we've had a lot of support. And I'm quite proud of the fact that we've been able to crowdfund this with donations and really kind of build it with beekeeper support in that way, rather than relying on federal grants or so on, although those are certainly nice to help pay the bills.
Yeah. Thank you so much. It was a great, great talk. Thanks so much.
All right. Well, thank you, everyone. I can go take a break and have a glass of water. And but everybody have a good night. Maybe in a year or two after the COVID vaccine is all spread around, I can take a break from this Pullman winter and come give a talk in person.
That would be nice. Thank you for giving up your evening for us.
All right. Well, have a good night, everyone.
Hey, you too.
Good night.
Thanks so much.
Good night.